All right, guys. Hi. The time has come for us to embark on the wonderful journey that is cellular respiration. Um, it is a long and convoluted journey that is three steps, really three and a half. Um, but we're going to go one step at a time and just make our way through it. So, first of all, cellular respiration, the goal is to convert. See what color the goal is converting sugars into free energy the cell can use. And to say this in a way that will make you maybe care about it, cellular respiration is the way that your food actually provides the energy to keep you alive. Um, It's also the way that the food that you eat eventually becomes like the gas that you exhale, which is like pretty crazy to think about that that's what's happening, but that's what's happening. Um, right here, you can see the diagram that I showed at the end of the last lecture that I did, which was just sort of an really basic overview of cellular respiration. And I want to start by kind of like recapping that, just using different words. And then we'll kind of dive into each step one bit at a time and go from there. So there are three main steps to this cellular respiration experience. They're over here. They're glycolysis, which is step one, the Krebs cycle, which is step two, and oxidative phosphorylation, which is step three. The oxidative phosphorylation step includes the electron transport chain and um, something called chemiosmosis, which we'll talk about when we get down to the bottom. And what I wanted to start by doing is just like a really brief overview of these steps so that when I dive into it, you'll have like kind of some idea what's going on. So the first step is glycolysis. Glycolysis is the step where glucose is split. Let me make that a little better. Split. Some ATP is produced. And glycolysis happens in the cytoplasm, which you also might see called the cytosol. They're the same thing, just the fluid in the floating around the cell. In between glycolysis and the Krebs cycle is something called the link reaction, which isn't technically like its own step, it's like a half step. And what happens then is the products of glycolysis, which is called pyruvate, is converted into something called acetyl CoA, which I talked about really briefly last time. But I'll say acetyl CoA formed. That's like the briefest synopsis of this. And that happens in the mitochondria. So now we get to the Krebs cycle. I'm going to just like highlight the link as being a separate thing, just so it doesn't get like kind of messy. And I'm going to label it as step 1.5. All right, so then we get to the Krebs cycle, which also can be called the citric acid cycle. They're both the same thing. In the Krebs cycle, what happens is the sugars that you ate are oxidized. So sugars oxidized to form some ATP, some CO2, and then also some electron carrying molecules, which I'll just write that for now. And this happens in the matrix of the mitochondria. And then finally, the last step is oxidative phosphorylation. I keep wanting to put a Z in there. And what happens in that step is electrons are used to generate a proton gradient and produce tons of ATP and also water. So I'll put that on there too. This happens in the inner mitochondrial membrane. I'm just going to write mitomembrane. I'm going to say slash intermembrane space. And that's kind of like the overview of this. So we have these 3 steps with sort of a half step in between steps 1 and 2 that results in our sugar that we ate being converted into carbon dioxide, energy, and water. And we'll like kind of come back to this picture at the end to summarize it. But for now, I'm going to start by diving into our friend glycolysis. So let's go over here. Glycolysis literally means sugar splitting. Okay, glycolysis is sugar splitting. And literally what's happening is 1 glucose, which is 6 carbons, is turned into 2 pyruvate, which are 3 carbons. In order to do this, it actually requires some ATP to be used up to split the sugar. So I'm going to go down here. We're kind of transporting ourselves back to biochem here just for a second while we talk about cell respiration and also while we talk about photosynthesis. But got to put yourself back in the mindset of sort of these like atoms and molecules being moved around. So it costs some ATP to break up glucose, and that's these. So this is called energy investment, which it says up at the top, but it consumes 2 ATP. And then the process actually releases 4 ATP. So this is energy payoff. So 4 ATP generated. And these ATP are generated using something called substrate level phosphorylation. And what that means is that a phosphate group is removed from substrate by an enzyme and then attached to ADP, oops, not ADR, ADP to make ATP. So literally what this means is that the phosphate is coming from the substrate. And the ADP as well as the substrate will fit into the active site of the enzyme. And that substrate level phosphorylation. The picture is showing that. So this one right here is showing that you have a substrate, which in this case is a sugar called PEP, which is an acronym. When the enzyme removes the phosphate from PEP and adds it onto ADP, the product that's formed is pyruvate. So that was the goal all along, is to make pyruvate from glucose. So now you have created a PP and you've created pyruvate. Those are the products of the enzyme. Um, Felt like there was something else I wanted to say about that, but I don't think there was. Okay. All right. So now we have generated 4 ATP, but at the beginning we used 2. So when it comes down to it at the end of the day, what we have done is we've produced 2 ATP net gain. We've produced 2 pyruvate, which are sugars, and we've also produced 2 NADH, which is an electron carrying molecule, going to be really important for later steps. And that is done, if I come up here, by adding electrons and also hydrogens onto a molecule called NAD+. You don't ever need to know like what NAD stands for. I don't even know what it stands for. But what is important to know is that whenever you add an electron onto a molecule, you're also going to add a hydrogen. And that's for charge reasons. And it's going to become important when we get to the electron transport chain later on. I'm just going to make a note that says when molecules pick up electrons, they also pick up the same number of hydrogen ions. So that's why NAD plus becomes NADH, because it picked up a hydrogen and also picked up an electron. So now it has a neutral charge. Okay, that is glycolysis. Something very important to say is that every single type of cell can do glycolysis, which I have highlighted, or I am highlighting right here. Every cell can do glycolysis, whether it is a bacteria or an animal cell or a plant cell, what have you. And also that glycolysis is not super efficient for producing energy. It's doable, but it's not the best. So keep that in mind as we go forth. Alrighty, let's go to step 2. So after glycolysis, we're going to move on to the Krebs cycle. Before we move on to the Krebs cycle, remember there's that sort of intermediate step that occurs. So I'm going to talk about that first. And then before I even go there, here's NADH and FADH2, which is important to talk about them before we really go any further. So NADH and FADH2 are both just electron carrying molecules. That's their main purpose. They're going to be really important for getting your electrons to the electron transport chain, which is where the most of your energy is going to actually be produced. So let's look at the Krebs cycle, and we're going to start with this link reaction, which is happening right here. This is the link reaction, which remember I labeled as like step 1.5. So here's what's actually happening there. In the pyruvate, a carboxyl group is being removed. When that carboxyl group is removed, it releases a carbon dioxide. Because if you look at a carboxyl group, it looks a whole lot like a carbon dioxide. So that's what's happening. At the same time, NAD plus is reduced to NADH. So just like I was mentioning before, same thing that happened in glycolysis is happening here. When something is reduced, it's adding electrons and hydrogen. So that's what's happening there. And then your acetyl-CoA is formed by adding an acetyl group to a molecule called coenzyme A. You don't have to know anything really more about that. So what occurs in step 1.5, which is the link reaction, is carbon dioxide is produced. Also produced is NADH, and finally acetyl-CoA. And acetyl-CoA is what you need to actually start the Krebs cycle. So, hooray, you have what you need. One other thing just to note is that this is happening in the mitochondria. Okay, so now we have what we need to start Krebs, which is our acetyl coenzyme A. Once we have that, what starts to happen is this entire just process that is catalyzed by all these different enzymes that you will not ever have to know the name of, probably. Chances are slim, um, to produce some different products. So, what you're going to produce is two carbon dioxide molecules, three NADH molecules, one ATP molecule, and one FADH2. So, let's see, do I have a place to write my products? No, but I'm not going to write them. So, one Krebs is going to yield for you two CO2, three NADH, one FADH2, and one ATP. Now, something that's really important is what I've written in this red bubble above this one, is that for each glucose molecule, you have to do the Krebs cycle 2 times because Krebs cycle starts with one acetyl-CoA. And one acetyl-CoA used to be one pyruvate. And one pyruvate used to be one half of glucose. So if you want to break down an entire glucose molecule, you have to complete this entire thing 2 times. So what I'm going to say here is for one glucose, times yield by 2. And that will give you your result. So for one glucose, the Krebs cycle makes 2 ATP because it has to complete that cycle twice, one for each pyruvate or one acetyl-CoA, whichever, same thing. Another thing to note is that whether you're a prokaryote or a eukaryote is going to affect where you are able to do this process. Prokaryotic cells can do aerobic respiration, they just don't have a mitochondria to do it in. So they're going to do it in their cytoplasm. So this is for prokaryotes. The matrix is for eukaryotes. I'm just going to abbreviate it. Most things are going to be doing this in their mitochondria. So if you ever saw a question that was asking, unless it's specified that it was a prokaryote, you would assume that it was referring to eukaryote for the question. Okay, and then here's like my favorite Krebs cycle fact, which is that at the end of the Krebs cycle, all of the glucose has been oxidized and actually is exhaled as carbon dioxide. So when you exhale, when you're exhaling right now, you're actually exhaling like the food that you ate earlier today or yesterday, depending on like when you're watching this video. So that is like a crazy amazing fact that your food, a lot of it is exhaled. The carbon, all of the carbon that was your food is exhaled. And if it was glucose, and the electrons are actually what's used to help generate the energy, the electrons that came from your food. So crazy, cool fact. Okay. So now we're ready to graduate to step 3. Up until this point, it's important for me to just mention that from the one glucose molecule we started with, we've only made 4 net ATP, which is really not very much. We made 2 in glycolysis because we consumed 2, and we've made 2 in the Krebs cycle. So we really haven't gotten that much energy out of the carbs that we ate. Now we're going to get them, the energy. All right. So the electron transport chain is just a series. Oops, don't want to be on highlight. I'm going to switch to a different color so it like stands. A series of proteins embedded in the mitochondrial inner membrane. So if I look at my like little diagram here, the first one is more like scientific drawing, which I'll talk about in a second. The one that I drew, I just want to show like kind of where this is. So inside the mitochondria is the matrix. And then the actual like proteins that are in the picture with the orange background are embedded sort of along this membrane. And then also worth it to note that like each mitochondria can have many electron transport chains. It doesn't only have one. So like there could be one there, and then there can be another electron transport chain like also existing. Um, it's like not one and done. And then of course this, I'll just label this that this is the inner membrane. This is the outer membrane. This space in between is the intermembrane space. And this is all going to be important here in just a second. Now, this is all, of course, for eukaryotes. So if you are a prokaryote or a bacteria, you're not going to have a mitochondria to use for this. So if you are a bacteria, you will use your plasma membrane instead. So the actual membrane of the cell could be used for this in a prokaryote. But now we're going to go back to eukaryotes because that's what we are. That's what we care about right now. Okay, so let's look at this picture and look at the goal. So the goal of the electron transport chain, which is similar to the goal of cell respiration on the whole, is to capture free energy from electrons and establish a proton gradient, an electrochemical gradient. And that's going to allow energy to actually be produced. So we've arrived here. Krebs cycle happened in the matrix. So our electron carrying molecules are, which were produced from Krebs, are already in the matrix, hanging out, waiting to be used. And here they are, NADH and FADH2. So I'm going to say that these are my carriers produced by Krebs. And they have now arrived at the electron transport chain. Now, what happens is that each of these proteins has increasing electronegativity. So they're more and more attractive to electrons. And that's how the electrons are pulled down the chain to eventually the end of the chain, which is the fourth protein subunit, where they're finally accepted by oxygen. So I'm going to make a note, I think, here. Yeah. That says oxygen is the final electron acceptor. Here. I'm not sure if that's spelled right, but spelling's not one of my gifts. Because it has high electronegativity. The oxygen accepts 2 electrons, and it also accepts 2 hydrogens because, like I said before, if it accepts an electron, it has to accept a hydrogen too. And that produces H2O, which is water. So accepts 2 electrons and 2 hydrogens to equal H2O. Hooray! All right, so that takes care of where the electrons went. But what happens also is that, remember, NADH and FADH2 are not only carrying electrons, they're also carrying hydrogens. So as the electrons are released, the hydrogens are also released, and they are pumped through these membrane proteins into the intermembrane space. So that's what's happening here. In the diagram, the orange space is the intermembrane space, which in my diagram is like the sandwich space in between the 2 membranes. Now, keep in mind that the other side of the intermembrane space is another membrane. And things that are charged cannot pass through the membrane because they're charged. And the membrane has these nonpolar fatty acids that make up the majority of the membrane. So what happens when the hydrogen ions are pumped through their channel proteins into the intermembrane space is that they can't escape. They can't get out the other side. They can't diffuse back through the membrane because they're charged. And also, I should note that the way that they're getting pumped through the membrane is using the kinetic energy of the electrons moving down the transport chain. That energy is powering the protein pumps so that the hydrogen ions can be pumped to where there's already a lot of hydrogen ions, so active transport. Finally, what happens is the hydrogen ions can get back through, but only through one special enzyme, the most special enzyme ever of the world, which is ATP synthase. And that's this like teardroppy look. That highlighter is not going to help me, but it's this ATP synthase. This diagram doesn't show it well, but ATP synthase is literally like a turbine revolving door where the hydrogen ions fit through, and that keeps the turbine turning. So the kinetic energy of the hydrogen ions flowing through is what actually allows an inorganic phosphate group to be combined with ADP to produce ATP. And that is like the big shebang of like, we did it, we made ATP, hooray, but we're making tons and tons and tons of ATP. That process of the hydrogen ions flowing back through ATP synthase is called chemiosmosis. Basically osmosis, but for chemicals, hydrogen ions. Together, the electron transport chain and chemiosmosis are called oxidative phosphorylation. If you remember before when I did glycolysis, I talked about substrate level phosphorylation. This is just another type where the energy to like combine those the ADP and the phosphate is not coming from the substrate. It's coming from the electron transport chain, oxygen accepting the electrons, and the proton gradient being formed. So now I'm gonna like do that in words on here. So what we have is hydrogen ions flowing through ATP synthase back into the matrix, which produces the ATP from the phosphate, and that is called chemiosmosis. I don't want to use red, I want to use green. And then also we have electrons being pulled from protein to protein because of the electronegativity. That is called the electron transport chain. And together, these processes are called oxidative phosphorylation, which is here. And this is what is going to create like max ATP. So this is super efficient at creating ATP. And all of this is possible because of the whole process working. So the electron carriers being produced in Krebs, the electron carriers can't be produced in Krebs if glycolysis doesn't produce pyruvate, and glycolysis can't produce pyruvate if you don't eat any glucose. So every step of the process depends on each other to work. So now I'm going to go back to the top and we're going to summarize this bad boy and then we're going to wrap it up. So here we are back at our picture. So here's what we have. This also shows our ATP yields, which is great. So first of all, for glycolysis, we are having 2 ATP net gain. We really make 4, but we use 2. We also are going to produce 2 ATP in the Krebs cycle. And then our oxidative phosphorylation experience, like I said, it's way more efficient. It's going to produce about 26 to 28 ATP. And I'll just say that the efficiency of this varies. At the same time, you're also producing these electron carrying molecules, which then are just recycled. So after they're used, they become NAD and FAD again, and they can go back and basically be reduced again. They're constantly being oxidized and reduced, and oxidized and reduced over and over and over again. And then I just want to look at our reaction really quick. So of course, this is aerobic cellular respiration, which I'll just make a note that this is aerobic cellular respiration. Aerobic, of course, means that it uses oxygen. And I'm just going to make a note of like where each of these things is either consumed or produced. These things on the left, of course, are the reactants. On the right, you have the products. The glucose is used in glycolysis. I'm going to do each step in a different color. So used in glycolysis. And I'm just going to make a note that it's completely oxidized by end of Krebs. The oxygen is used in the electron transport chain as final electron acceptor. Then our carbon dioxide is produced my pen thickness, my OCD is kicking in. Produced in Krebs and also a tiny bit the link reaction. The water is produced in the electron transport chain when oxygen accepts 2 electrons and 2 hydrogens. And the ATP, do a different color, because this is produced in all 3 steps and it will be 2 in glycolysis, 2 in Krebs, and that's per glucose. And then 26 to 28-ish in electron transport chain slash chemiosmosis. And there you have it. So that is aerobic cellular respiration. 3 steps to make a ton of ATP, keep you alive, break down your food. Magic. Thanks for watching.